building. My office happened to be also in this particular building, so it's very nice. When there is sun, it's very nice. That's very rare. The work I show, because I'm speaking in a conference in India, the work I show actually, the data part was generated in India, and this is happening in collaboration with me, so you will see as I go along. As we are targeting to the younger members of the audience for right reasons, so I'll also start with the basics. So we all know the central dogma of molecular biology, and now we also know that that is insufficient to explain all the complexities we see. We know all the epigenetic regulations by non-coding RNA, and you have been hearing since yesterday, I guess, which I missed, about all different ways these three layers, these three primary points of the central dogma can be modulated. So you've heard about DNA epigenetics a lot. I'll talk to you something on RNA, so epitranscriptomics, and the protein also gets a lot of modification. The central dogma of molecular biology is not as simple as we once thought. And particularly when we look at the number of genes in humans, which is about 25,000, that don't explain the complexity that we have as an organism. Just for a comparison, E. coli has 18,000 genes, right? Which is known as C-value paradox. You probably know from your textbooks. Now, at the mRNA level, the diversity increases by splice isoforms. At the protein level, the diversity increases further, right? By many different modifications, epitranscriptomic modification as well as post-translational modifications. So the complexity increases from genome to proteome all the way, and I'm going to talk about complexities in the middle. This summarizes what all I do. I try to put in the context of why I do what I do. Pretty much everything this community tries to do is G2P, which means genotype to phenotype. So we see a phenotype and we hope that we'll find a molecular signature which will explain the phenotype. Right? That's the summary of everything we are trying to do. If you think it from a point of view of information flow, how the information flows, DNA is like the blueprint. Right? The blueprint leads to RNA, the RNA leads to protein, and all the layers of information in addition to the environmental exposure leads to the phenotype. So for a molecular signature to accurately predict the phenotype, that means the molecular signature's penetrance, that we call in genetics, has to be very, very high, so that all other layers of information cannot influence. Right? So I work on molecular variation. The change fascinates me essentially at the molecular level. I work at the entire spectrum. Today's talk is focused on that particular layer of information. How does things change in that particular layer, and how that can explain phenotype? Okay? Part of which will be focused on normal samples, and then towards a later part of the talk I'll show you how it relates to cancer epigenetics and cancer biomarkers. If you think at the RNA level, the RNA contributes diversity to the human transcriptome in many different ways. I will not go to the detail of all different ways, just there can be exon skipping, intron retention, alternative splice isoforms, both are 5' and 3', and mutually exclusive exons, which is the primary splice isoforms, and then alternative polyaccumulation signals. All these ways that RNA can increase diversity in our cells and tissues. It can also do in two other ways, which is the epitranscriptomic changes. One is RNA editing, which I will not talk about today, but that's also epitranscriptomic change. And the reason I'm focusing on epitranscriptomic change is because this is somewhere RNA does not encode the genome accurately. There are changes. What is the change? Most common type of editing is A to I, which looks like A to G. And DNA base, which is an A, in RNA gets changed to G, and that happens in a tissue-specific manner. So that will produce diversity. But my today's focus is a different kind of transcriptomic change, which is fusion transcripts. Because this is a cancer-focused conference, you probably all know the Philadelphia chromosome, the BCR-able fusion gene. So fusion transcripts are very well known, at least for that particular case, as cancer aberration. Right? But what I'm asking is, it only happen in cancer or also happen in normal situation? Right? Particularly in the human brain. I'll explain why brain is of particular interest. So what is fusion transcript? The two genes, they're distinctly two different genes, can be on the same chromosome, can be on two different chromosomes. Either at the genomic level due to translocations, as been shown by other speakers, or at the post-transcriptional processing level, pieces of gene A and pieces of gene B can get fused together, and that can lead to an aberrant molecule which can have a function. Sometimes it can also be noise, transcriptional noise. Every fusion transcript need not have a function. Transcription is a very noisy phenomenon. So that's something you need to keep in mind. Another thing I want to point out, that between these two types of RNA modification, epitranscriptomic modification, there is another fundamental difference. This RNA editing is a tightly controlled enzymatic reaction. In other words, it's a non-random change. Where a fusion transcript is more random, it's more stochastic. Now, why we focus on the human brain at the RNA level? On the x-axis here, you see an age. So this is birth. This is at birth. So before birth and after birth, and this is like an infant, first year of life. If you look at the synapse development which happens in the brain, look at the light blue color. So it goes up and it peaks at about birth, and then it tails. So this particular fusion is what I'll talk more about. Before that, sorry, I forgot about this. Before that, we also had a question that how we, as I said, they are stochastic, they are probably transcriptional noise. So we wanted to understand, is there any evidence that these fusion transcripts leads to any protein molecules, peptide molecules? So we did an extensive proteomics analysis in the available database, and we did find that about 20% of the fusion transcripts we found to have a translated product in the proteomic database from the brain. So we found the peptides that spans the fusion junction. Right? So we found those proteins. So it looks like a minority fraction of these fusion transcripts can lead to protein. Whether this protein will create a problem or not, we don't know. You will see an example. So this particular fusion we picked up because this was proposed to be a cancer biomarker, like the Philadelphia chromosome, as said in the beginning. So we wanted to test that in our setting. So what is this fusion? So fusion between two different genes, CANCEL1 and ARL17, and this part gets inverted and comes here, and this part comes there, and this particular fusion happens. This is quite well known, well characterized. We found that fusion in 16% of normal brain tissues. That was the first surprise. So earlier, all reports suggested that this is a cancer biomarker, such as the Philadelphia chromosome. But in our case, first we found in normal brain tissues. Also, we found DNA level evidences. So these are DNA level translocations that is causing this fusion, not at the RNA level. And then, with collaborations with AIMS Delhi, we looked into brain cancer. Why? Because our data so far was coming from the brain. We had reasons to think that neuronal cells will have more fusion events. And so we wanted to see whether this particular fusion as a candidate has a biomarker potential for glioblastoma multiforme. Now, glioblastoma multiforme is the most aggressive type of brain cancer. It's a very, very poor surviving prognosis. And so I guess this audience knows about GBM. We had only 9 samples from AIMS Delhi, and those 9 samples, I think 7 of them showed the presence of this particular, 7 of them showed this particular fusion. So while we found only 16%, although it's a smaller sample size, 16% in normal brain, so in our sample, 78% of the GBM sample had this particular fusion. So we thought that indeed it will be a biomarker. However, we then went into the details of the DNA level reorganization, and we found out there are 2 different haplotypes for this fusion at the DNA level. The H2 haplotype is very rare in Asians. The H1 haplotype is much more common in Asian, much rarer in Europeans at the genetic level. We realized that the way we were doing the assay at the DNA level before was actually targeting the H2 haplotype. Now when we moved to the H1 haplotype, what we found that our sample, almost all of them at the H1 haplotype, which means the 16% was a lower estimate. So when we looked at the specific H2 haplotype, we found that fusion product is present in 7 out of 10 normal samples, so much higher proportion. Very similar to GBM. So it's 70% in both control as well as brain cancer patients at the DNA level. So at that point, this did not look like a promising biomarker for GBM, this fusion transcript. It looked like it's happening all the time, so it's present in any kind of brain sample you can pick up. But then when you look at the RNA level, this is where the precision genomics or precision medicine comes into picture. And then we look at the RNA level. First, we looked at the RT-PCR of fusion transcript between frontal cortex, corpus callosum, and GBM samples, and we found the GBM sample to have a much lower expression of this particular, although the number frequency is the same, expression is much lower for the fusion transcript. But if you look at the parent genes that got fused, parent gene is opposite. It's much higher in the GBM samples compared to the two parts of the normal brain. And the other parent gene, the frontal cortex, is higher than either corpus callosum or glioblastoma multiforme. I should mention here that if you think of the correct cell type as a control for GBM, because GBM is an astrocytic tumor, corpus callosum actually is a better control cell type than the frontal cortex, than the neuronal cells. So we compare more with corpus callosum when we look at GBM. So when you compare corpus callosum, the GBM is 5 times upregulated in the normal, or in other words, it's 5 times downregulated in the GBM, the fusion transcript. CANCEL1 is upregulated in GBM, again 5 times, and ARL17 expression in CC and GBM is very similar, comparable. So looks like that the fusion transcript is competing. So basically what I'm trying to say is the parent genes are also present in the system. The CANCEL1 and ARL17 is also present in the system. The fusion gene is also present in the system, where all 3 of them are present, because some of the copies are gone, that is truncated and moved to fusion transcripts. There is a competition, right? So although at the DNA level you cannot use this in Indian population as a biomarker, but the RNA level you probably can. RNA level, they look like significantly differently expressed between the normal tissue and the cancer tissue. So summary, fusion transcripts are abundant in neurons compared to astrocytes. So first of all, fusion transcripts is not only a cancer-specific phenomenon, it happens all the time in all the tissues, I presume. We have looked into the brain, but I'm sure it happens in other tissues as well. Fusion transcripts accumulate with age in the human brain, and we think this is a way of post-mitotic cells to introduce diversity. Pathway analysis of genes harboring fusions has revealed an enrichment for genes implicated in the process involved in synaptic plasticity. Brain cells and tissues have contrasting spectra of fusion transcripts in terms of inter- and interchromosomal events. Random splicing is the possible mechanism for trans-splicing events, thus retain splicing signature. I did not show you this data. Indians have the H1 haplotype at the locus with allele frequency of 70%. This is a novel finding, it's not known before. And this particular fusion is a similar DNA level frequency, but drastically different expression level between normal and cancerous brain, which means this can become a precise biomarker for glioblastoma multiforme particularly. The work has been done by Bharti. This is her PhD dissertation. Bharti is now doing postdoc in the US. This is funded partly by DBT, and these were clinical collaborators from AIMS Delhi, and this has been published a few years ago. Neurons. So these are post-mitotic cells. That means cell division cannot be a mechanism to introduce diversity in this particular cellular population. So it needs to have a different mechanism to introduce diversity, right? That's the reason we are interested to study normal brain and to ask the question, does fusion is one way that these post-mitotic cells introduce diversity in them? And the reason we thought that, made that hypothesis is stemmed from this particular paper. So this paper showed this one particular fusion, here, which can happen in both DNA level as well as at RNA level. So I'll repeat here, same fusion, two different genes, one on chromosome 7 and one on chromosome 17. It can create the fusion both by DNA translocation as well as by post-transcriptional processing. What is the difference? If you have it at the DNA level, which makes it constitutional, it leads to cancer. If you have it at the RNA level, which can be tissue specific and not constitutional, it leads to growth advantage. So this triggered my interest that same mechanism, same outcome, but depending on what extent it is present in the body, can lead to a different phenotypic outcome, which one case is cancer, other case is essentially a controlled growth advantage. If you think of cancer as an uncontrolled growth advantage, so if you have a constitutional level, it's uncontrolled growth advantage, which we call cancer, and the RNA level, this is a controlled growth advantage. So a post-transcriptional processing can lead to advantages to the cell, which is other ways detrimental. So this is what was our key paper to start our work. This is a work pipeline which I'll not go into details. So essentially this is a genomic project. We did RNA sequencing, and what we looked for, so it's a paired end read, those of you know what I'm talking about. We looked for reads that do not map as it should be, right? So discordant reads. So two pairs maps to two different locations. So that way, and then we found breakpoint covering reads, and that we characterized fusion transcript, and we of course validated them as well. So what did we study before we went to cancer? From Nimhan's Brain Bank in Bangalore, we collected brain tissue samples, but what did we collect? We collected specifically the frontal cortex, which is neuron rich, and corpus callosum, which is neuron deficient, astrocyte rich, from the same individuals. So 10 pairs of frontal cortex and corpus callosum was collected, and there we did RNA sequencing. Along with that, we also analyzed RNA sequencing data of 49 cerebral cortex RNA sequencing from the public domain. And we also have analyzed single cell RNA sequencing data publicly available of 131 single neurons and 62 single astrocytic cells. Again, this is the workflow. As I told you, we looked at the reads that do not map where it's supposed to, and we also found reads that are spanning the boundary of the fusion molecules as a proof that these are indeed fusions, and these are all the QC steps. First of all, as you all know, anybody who dealt with the next generation sequencing data, that you have to do a lot of quality control. What you are essentially seeing is that the yellow bars are before filtering and the gray bars are after filtering, so you remove some of the reads because of poor quality. First, what we saw, so CC stands for corpus callosum and FC stands for frontal cortex. So how you should read the data is that 150 means this one individual. From one individual, you are looking at fusion per million reads, number of fusion per million reads, and that goes to the CC and FC are differently populated. So number of fusion overall is more in frontal cortex than in corpus callosum in the same individual. So we are looking at two different tissues from the same individuals, and we are seeing different numbers of fusion transcripts, and those are more in the neuronal rich samples with the frontal cortex. And this is PCA analysis to show that based on fusion transcripts alone, you can actually separate out the corpus callosum and frontal cortex samples. Then we ask the question, how does it happen in the single cells? What we are seeing in corpus callosum, which is representative of astrocytic cells, and in frontal cortex representative of neuronal cells. If you look at the single cells, neurons and astrocytes, how does it look like? It's even more profound. You can separate them out much better than their tissue representatives based on the fusion transcript between astrocytes and neurons. I told you that frontal cortex samples from the same individual showed higher fusion transcript than corpus callosum samples from the same individuals. In single cells, we also found the same thing. Overall, neuronal single cells had more fusion transcript than astrocytic cells. These are, of course, not from the same individuals, so it's not exactly translatable data. So how does it distribute? So now you are seeing fusion reads, and it's now significantly different in single neurons, and this color key shows that this is not because these genes that are fused in neurons are not expressed in astrocytes. This color key shows that these genes that are fused in neurons and astrocytes are expressed at the same level in the two tissues. The parent genes, fusion gene expression you do not get in this kind of heat map. The parent genes are expressed at a pretty similar level. So absence of the RNA transcript is not the reason that you are seeing a lower number of fusions in astrocytic cells. So astrocytic cells are just have less number of fusions that are happening inside them compared to neurons. So why do neurons have a preference for high number of fusions but astrocytes don't? So can we relate abundance of fusion transcripts to the regenerability of the cell? If you remember, I said our interest of going to neurons were they are post-mitotic, and we are hypothesizing that can fusion transcript be a way how neuronal cells create diversity amongst them. So for that, what did we do? We looked at adult neural cell, fetal neuron. These are single cell. This is from publicly available data. And same way we analyzed the data. And we also did a frontal cortex analysis between adults and newborn individuals. This is tissue level. So tissue level and single cell level, we looked at different age groups, essentially fetal and adult. What do we find? We find in adult neuron, number of fusions are significantly higher than in the fetal neuron. And in adult brain, the same observation. Significantly higher than the fetal brain. So we found that in normal brain, without any brain disease, brain cancer, any other brain disease, fusion transcripts tend to accumulate with the aging brain. And more interestingly, when we looked at the pathways where these fusion transcripts maps to, in the adult brain, they lead to long-term potentiation, and in the fetal brain, they map to long-term depression. This depression is not the mental health condition. This means depression of the synaptic movement, neuronal projection, right? So if you align them with the neurogenesis, we know in the fetal brain there's a lot of culling happens, a lot of neurons die, right? So that kind of matches that the different set of genes are getting fused in between adult's brain and the fetal brain, showing different pathway enrichments. How they are distributed? Very surprisingly, at this we cannot explain why. In the tissue level, we find intrachromosomal events. So the fusion are happening between two genes that are in the same chromosome. But in the single cell events, we are finding more interchromosomal events. So which might mean that lots of them are transcriptional noises, stochastic, and at the tissue level we are getting a summary, but we cannot explain this phenomenon, why this is striking the different way of organization of fusion transcript. But we do find overlaps. So these are the recurrent events that are found in both single neurons as well as the frontal cortex tissue level, cerebral cortex tissues. So that gives us the confidence that all of them are not noise, not random. So there are some functional relevance between these fusion transcripts. Now I'm coming to the part where we'll connect to cancer. So we found a few, we validated a few of the specific fusions. We validated means we found a PCR, we